Welcome to the Bio and Beyond podcast. I'm your host, Satwik Nath, and today we're diving into the world of biological engineering. Please join me in welcoming our distinguished guest, Dr. Anders Hansen. He is from the Department of Biological Engineering at MIT. At MIT, among many other things, he teaches instrumentation and measurement for biological systems and fields, forces, and flows in biological systems. His area of research is cancer biology and instrumentation and measurement. He is the recipient of awards and honors like the NIH Director's New Innovator Award and the Pew-Stewart Scholar in Cancer Research. At the Hansen Lab, he and his team work on understanding the interplay between genome organization and regulation of gene expression in mammals. Without further delay, let's dive right in. So, would you like to tell us a little bit about how you got to where you are today?
How far back should we go?
However far back you think is relevant.
Okay, well, it's always hard to say when things actually began. Well, briefly, I'm from Denmark, so I grew up in Denmark and spent the first 2 decades or so of my life in Denmark, and then in high school, I got very interested in chemistry, so I wanted to study chemistry at university. I started in Denmark, but eventually moved to the University of Oxford in the UK, where I studied chemistry and studied especially organic chemistry and did research in organic chemistry. And I think chemistry is incredibly attractive because, especially in organic chemistry, you only need to learn a few principles and then you can, by logic, deduce reaction mechanisms. So there's almost no memorization. But it is a very mature science. So a lot of the key foundational questions have been addressed. So during my master's, I got increasingly interested in biology because biology is kind of the opposite. Once you really dig down, there's almost nothing that we truly understand in biology. Take the most basic process like transcription or translation, and there are hundreds of labs that study it still, and we don't really know how it works. So that's very exciting. And so I wanted to shift to biology, but taking very quantitative approaches and molecular approaches like I learned in chemistry. So I did my PhD in chemistry and chemical biology at Harvard, which is a very flexible program. So I actually ended up doing more systems biology, trying to understand how cells receive and transmit information using amplitude and frequency encoding, a little bit like we use in AM and FM radio. And then I pursued a postdoc in biophysics at UC Berkeley, really single molecule biophysics. And then I started my lab just before the COVID pandemic at MIT in biological engineering. And biological engineering is an interesting discipline, and the department at MIT has really helped define this discipline. And it's a little bit different than biomedical engineering, which of course is also a very broad discipline, but it often has some focus on developing, say, devices for to help with biomedically relevant measurements and such. Whereas biological engineering, as MI, at least as MIT has defined it, is more focused on taking the tools of engineering and then really applying them broadly across biological disciplines to solve problems and develop new technologies. So this could be biomedicine, but it could also be agriculture, energy, climate change, and such. And so it's an exciting department to be a part of, and it's a very broad department as well. And people in the department come from many different backgrounds, both engineering backgrounds and non-engineering backgrounds.
Yeah, so one thing which I always find is interesting is whenever people come from outside of the country, they always have a, let's say, different experience in education. So would you
That's
like to
right.
tell us a little bit about your experience? Because I'm sure it's interesting.
Yeah, well, so I mean, one thing I can say is that the chemistry program at Oxford is very focused. So I only had 3 courses during my entire undergrad. I had physical, well, I had math, but other than that, it was physical chemistry, organic chemistry, and inorganic chemistry, and nothing else. And so by virtue of being extremely focused, you can go very, very deep. But you also then don't have as broad as an experience as you do in colleges in the US, where you take humanities classes and lots of broad classes. So it was very focused, which I think there are pros and cons to it. If you end up ultimately pursuing a career outside of chemistry, probably it's good if it's a little bit less focused. I think I took 3 or 4 separate courses on lanthanide coordination chemistry, which is a lot of lanthanide coordination chemistry. So yeah, those are some of the pros and cons.
Yeah, it's always interesting to hear about people's experiences outside of the country. Now, another question I had is there like a moment which really got you interested in biological engineering? Like, is there something you did which made you think, this is what I want to do?
Yeah, so I think one thing that that attracted me to biological engineering and also to MIT is that many places have a little bit of a divide between people who develop technologies and people who answer questions. So many places you do one or the other. If you answer questions, you might be interested in, I don't know, how does translation work, or how is biogenesis of mitochondria regulated, or things like that. And then you're very focused on answering the questions. And you might also have labs that develop technologies, and then they develop one technology, another one, another one, another one, and push lots of technologies out. And what I think you can really do to a unique extent here in biological engineering is both. So you start with the questions, but then you also develop the technologies, and then you use those technologies to answer the questions that haven't been addressed because the technologies weren't available. And I actually also think that combining the two is perhaps a great way of doing technology dissemination. Because if you just develop a technology, it tends to go to a technology journal, and other labs that might use it might not see it. But if you develop a technology and use it to answer a question that's of high interest, then other people can see how you can use that technology and become interested in it. So I think at MIT and MIT Biological Engineering, there's really a unique appreciation for doing both of those, and that's one thing I really enjoy about MIT.
Yeah, that sounds really interesting. Um, so another question I had is, could you please tell us a little bit about the work you do on genome organization and regulation?
Yes, absolutely. So why are we interested in genome organization? So the motivation is to understand how gene expression is regulated, right? I mean, you and I both started as a single cell many years ago and developed into a full human. And that this developmental process was encoded in the genome of the cell from which we came and involved a very carefully controlled spatiotemporal developmental program, expressing the right genes at the right time to the right extent. And now, of course, as adults, we are composed of many, many different cell types, muscle cells, liver cells, etc. They all have the same DNA. And so this really requires very careful control of gene expression. Moreover, misregulation of gene expression is a major cause of disease. To a first approximation, cancer means expressing growth genes too much. And so understanding how gene expression is regulated is in my view, one of the most foundational and most interesting questions there are. But it's tricky in humans, because if you take an organism like yeast, about give or take 70% of the genome encodes for proteins. So it's overwhelmingly coding. And if you take the human genome, about give or take about 1.5% of the genome encodes for proteins. 98.5 or so is non-coding. And so we have these gigantic stretches of DNA that don't have any genes on them. And it turns out they're used for regulation. And the pieces of DNA that control expression of genes in mammals can be extremely far away from the genes that they control. And they're called enhancers. And the genome is very, very dense. A typical megabase, so that's 1 million base pairs, will have on the order of 5 to 10 genes and 50 to 100 enhancers. And so this raises the question of how does the cell, with all these enhancers scattered throughout the genome, make sure that the right enhancer interacts with the right gene? Because we know that in order to activate expression, the enhancers has to loop to the genes. So how is this process regulated? And this is a foundational question that we don't know the answer to. Enhancers were discovered in 1981, and we still don't know the most basic questions of how do enhancers find the right gene and how do they interact. And so this requires us to understand the 3D organization of the genome because it's a 3D problem, right? We have the 1D sequence now that we've sequenced the genome, but we don't know the rules that govern whether this piece is going to loop over to that piece or another piece. And so that's why understanding the 3D organization of the genome is really important. And one of our premises relating to what we just talked about as well is that the reason why we don't know this is because we haven't had the right technologies. So for example, what you might want to do if you want to understand how looping happens is be able to watch this process in real time. So one of the things we've been developing are new super resolution live cell microscopy methods that allow us to fluorescently label specific pieces of DNA inside the cell, and then we can watch how they loop and find each other in real time. And then we can also see how do looping events result in transcription or not, and really get at cause and effect, right? Because if looping happens first and transcription comes afterwards, looping was probably causal. So that's one motivation. We've also been developing some 3D genomics methods for mapping genome structure at high resolution. So we develop these new technologies and then we apply them to systems of interest, both for understanding the basic mechanisms and also for understanding misregulation in disease.
This all sounds extremely interesting and extremely complex. One thing that I was wondering, since it sounded so complex, are there like any difficulties that you experience while working on this?
Every day. Yeah. No, I mean it is extremely complex, right? And I mean if we just take a step back, right? A mammalian cell has on the order of 20 to 25,000 genes that encode for proteins. And we don't know what most of those proteins do, right?
Yeah.
So you're studying a cell that's full of stuff that you mostly don't know what is and what it does, right? It is very complex. That said, there are a few sort of basic principles that have emerged. There is, in terms of genome organization, it seems like to a first approximation, we can sort of reduce it down to two major mechanisms. Let me see. This is a little bit of an ad hoc prop. There's a mechanism, so that let's say this is a piece of DNA here, right? There's a mechanism of loop extrusion. So there are these protein complexes that are not quite rings, but look a little bit like rings that land on DNA and can extrude loops like this. And then there are boundary proteins that tell it where to stop. And so this is one way of saying, I want these two pieces of DNA to interact, is to make sure there are boundary proteins next to them to hold them together. So this mechanism is called loop extrusion. And then the other mechanism is sometimes called compartmentalization or affinity. If you imagine that instead of having the DNA being just all the same, the different segments of the DNA, maybe there's an A segment here and a B segment here, and that largely corresponds to activity, whether the genes are on or off. And if those have separate affinity for each other, the A segments will come together and the B segments will come together. And that's through affinity-mediated interactions, and that's called compartmentalization. So while it is incredibly complex, those are sort of the two major mechanisms that we know of. There may very well be other mechanisms as well, but that's also why we like to go bottom up and try to understand what are the basic biophysical principles, and then build complexity one layer at a time to really understand the general principles and have some conceptual frameworks.
Okay. So that's all really interesting, but you also work on cancer biology, correct?
That's right. Yeah. Well, and disease more broadly, but yeah, we have several cancer projects.
Yeah, so could you tell us a bit about this?
Yeah. And I mean, again, cancer is an incredibly complicated disease. There are many, many different types of cancers that have many different properties. But highly simplified, cancer is a disease of too much growth, right? That's you have lots of cells as an adult that are not supposed to divide anymore. And if they start dividing too much, you can build up to a tumor and you can get cancer. And whether or not a cell chooses to divide or chooses not to divide depends on growth genes. So there are a lot of growth genes that promote growth. And so what you see is that in many cancers, you have growth genes that are not supposed to be active that are active. And there are many different causes of cancer, but a frequent cause of cancer is misregulation of 3D genome structure that leads to overexpression of growth genes, and that then leads to cancer. And so in our cancer biology work, we are often focused on specific situations where, let's say, you have a growth gene here and you have an enhancer here. And in normal cells, they don't talk to each other, but there's some sort of misregulation of 3D genome structure that allow them to talk to each other. And then oftentimes the genetics will be clear, but the mechanisms won't be clear. And then we try to go in and use some of the technologies we've developed to understand what are the mechanisms supposed to be like in a cell that's not cancerous? What is it in the cancer that causes them to interact in a way they're not supposed to do? And once we understand those mechanisms, are there some things that we can do about it?
Could you tell us a little bit more about the technologies you work with?
Yeah, so we work with a ton. I mean, I think that the two main ones we've recently innovated is super resolution live cell imaging, so and high resolution 3D genome structure mapping. So super resolution live cell imaging allows us to watch inside of living cells how different pieces of DNA move and find each other, and then at the same time watch gene expression in real time. And this allows us to ask these questions of how do enhancers and genes find each other, and how do they turn on a transcription. And this is a foundational sort of textbook level gene regulation question, but it's also important in disease. So let's pick a specific example. So this is some work that's mainly been led by Brad Bernstein's lab at Dana-Farber, but we collaborate with them to apply some of our technologies. They found that in certain cancers, there you have an enhancer, you have a gene, and then you have an insulator that prevents the enhancer from talking to the gene. And then in these cancers, epigenetics, specifically DNA methylation, inactivates the insulator that sort of blocks the interaction. Now the enhancer can activate the gene, and the gene is a growth gene, and that then leads to the cancer. So that's true in IDH1 mutant glioma, which is a type of brain cancer, and SDH deficient GIST, gastrointestinal stromal tumor. And they're actually, based on understanding the mechanisms, they're actually doing clinical trials now. I hope I'm allowed to say that, but yeah, they're finding some exciting things that may really make a difference in patients' lives.
That's really interesting and sounds very helpful to society. One thing I was wondering is, what would you say would the future of your field looks like? Like, how
Yeah,
would AI interact with your field, and how would it affect the future?
well, how would AI affect the future? I don't think I can address that. What does society
Yeah, I
look
was
like in
asking how.
2134? I would love to know. Yeah,
Yeah, how would your field affect the future?
that's right. Yeah, no. It's a very good question, right? And AI is moving so fast and seems to be exponentially improving that prognosticating about the future is difficult. But I think so. We're using AI in some practical ways. So we do a lot of microscopy, and so then you want to have movies, images, and convert them into some insights, right? And AI is very powerful for image processing. So there are various image processing problems that we use machine learning approaches to solve. AI has also had a big impact on being able to predict how elements in the genome function. So the human genome contains about 3 billion letters, and the information content is actually, it's relatively low in the sense that it's not like the letters are always exactly the same for the exact same element. Like it's not like every single promoters have exactly the same sequence, every single enhancers. And so there are all these questions of how do you interpret the genome and how do you predict what this piece of DNA is going to do and that piece of DNA is going to do, that AI has proven to be quite good at. A specific thing that we're doing in my own group is that we've developed this technology called region capture micro-C that allows us to map 3D genome structure at about 100 times higher resolution than what was possible in the past, which is fantastic, but only for small subsets of the genome. And so we can't do it for the whole genome. It would cost hundreds of millions of dollars. And so what we're using machine learning for in that case is let's do these ultra deep mappings of a few regions, and then let's use machine learning to learn how to go from low resolution to high resolution and then impute the rest of the genome. And I think ultimately, and this is probably not too many years away, I think we can use machine learning to predict how to sort of computationally do a lot of experiments, a lot of perturbations, a lot of tests, and then prioritize which ones we are then going to confirm experimentally, right? Because right now the way we do things is that we read papers and we think about it and we have a hunch and then we do some experiments to test it. But I think and that works very well. I mean, much of the progress in science and engineering comes from that approach, but it's of course not as fast. So I think in the future, hopefully, we'll be able to ask a lot more questions and get approximate answers using machine learning and AI, and then go in and test them. And in the slightly more distal future, right, you could imagine AI approaches predicting your likelihood of disease and what will be helpful for you and so forth. So it's very exciting times and things are moving very fast.
Yeah, I think the applications of AI in the future are limitless, and that what we could do compared to now is crazy. Like, I feel like we could solve many of the problems in the future with AI that we are having right now.
That's right. That's right.
Now, one more question I had is, now let's take a step back from AI, because I wanted to know more about how you work in the lab. So what is a skill that you use in the lab that you would say most people wouldn't expect you to use in the lab?
Yeah, that's a good question. So, I mean, these days I'm sadly not physically in the lab anymore because there are too many meetings and too much teaching and too many grants to write and forms to fill out and such. And so the real research, so to speak, is done by the graduate students and postdocs in the lab. And so I think one of the most challenging aspects of being a PI, a principal investigator, is keeping track of everything that's going on and the switching. So I think that's maybe not as exciting. I don't know if you're looking for a very specific technical skill, but honestly, I think that is the most challenging aspect of academic science as it is today.
It's always interesting to see what skills people use that
Yeah.
normal people wouldn't expect for someone such as you to be using. Anyways, the last question I had is, what would you advise to high schoolers who want to pursue biological engineering in universities?
Yeah, it's a good question that I unfortunately am actually not the best person to ask, because coming back to what you asked at the beginning of what are some of the unique perspectives you have from having studied outside of the US, One of them is that I didn't go to high school in the US, and so I am not as familiar with high school. I could give some advice to Danish high school students if you have any in your audience, but that's probably a relatively a niche audience. So I'll keep it quite general, and I think so I'm on the graduate admissions committee, so the PhD committee. And what we're looking for there, so that the people who apply have gotten their bachelor's degree. What we're looking for there are people who have training both in math and engineering and also biology. So I think if you are interested in pursuing biological engineering or biomedical engineering, I think the key is to be able to bridge between the biological sciences as well as the engineering, quantitative, and physical sciences. So having a background both in biology and math, physics, chemistry, and such, I think will be great.
Okay, so is there anything else you would like to add before we end?
No, this has been great. Yeah, thanks for having me on.
No problem. Well, as we wrap up this insightful episode of the Bio and Beyond podcast, I want to extend our sincere gratitude to Dr. Hansen for sharing his wealth of knowledge and expertise. His commitment to the field of biological engineering and his passion for academics is truly inspiring. If you enjoyed this episode, stay tuned for more captivating conversations at the intersection of biology, medicine, and technology. Until next time, this is Sawik Nath signing off from the Bio and Beyond podcast. Remember, the exploration never ends. There's always more to discover.